Chagas disease is the clinical condition that ensues chronic infections by the flagellate protozoan Trypanosoma cruzi. The disease attacks the heart of chronically infected people several decades after acquisition of the infections. A cross-kingdom model system shows that chicken eggs are permissive to the Trypanosoma cruzi infections during the first week of embryo growth. The experiments show that infection-free chickens retaining the parasite kDNA into their genomes die of Chagas heart disease. Excellent housing, food, and water supply secures animal welfare. The Trypanosoma cruzi trypamastgoat swimming forms are obtained from tissue culture. Transilluminate fertile chicken eggs to show their bubble. Control eggs do not receive parasite inoculation. Mock control eggs are inoculated with 10 microliter of culture medium. The whole is sealed with adhesive tape. 100 virulent Leishmania brasiliensis pro-mastigoats are inoculated in the air chamber of fertilized chicken egg. 100 virulent Trypanosoma cruzi triple mastigoats in 10 microliter of culture medium are inoculated in the air chamber of fertilized chicken egg. The eggs are incubated at 37 centigrades at 65% humidity for 21 days with gentle rotation each 30 minutes. The chicks hatch equally from Trypanosoma cruzi inoculated eggs and from mock controls. The chicks grow to adult life. Blood samples are drawn from wing veins. Blood is transferred to 15 ml conical tubes. Mononuclear cells are separated by centrifugation upon Fickel-Hypake gradient. Wash off white cells pellet and discard separation medium. Genomic DNA is extracted from blood mononuclear cells. Set 1.5 ml conical tubes in racks according with PCR design. PCR is made to amplify T-cruzi nuclear DNA, nDNA, and kinetoplast DNA, kDNA, using specific primer sets. Set reaction tubes in the thermocycler. After run, take tubes back from thermocycler. Analyze the PCR products. The PCR products are separated by agarose gel electrophoresis. The DNA bands in the gel are transferred to a positively charged nylon membrane. Hybridization is made with specific radioactive labeled probes. To separate roles played independently by the parasite persistence and by the autoimmunity in the pathogenesis of Chagas disease, we use a chicken model refractory to the Trypanosoma cruzi infection, except during the early embryonic growth prior to the development of the immune system. The chicks that hatch from Trypanosoma cruzi inoculated eggs are infection-free, but the parasite mitochondrial DNA, kDNA, is retained in the body. Only chickens hatched from Trypanosoma cruzi inoculated eggs show kDNA bands. The nDNA is not present in the chicken genome. Chicks hatched from eggs inoculated with spermatogotes of Leishmania brasiliensis do not retain the mitochondrial kDNA minicircle sequences in their genomes. Lane 1, blank. Lane 2, control DNA. Lanes 3 and 4, mock DNAs. Lanes 5 to 14, DNAs from chicks hatched from Leishmania brasiliensis infected eggs. Lane 15, Leishmania brasiliensis amplicon, formed band 750 base pairs, obtained with LB3 and LB5 specific kDNA primer sets. TEL-PCR. A checkerboard scheme depicts the Gallus-Gallus primers that were deployed after 3 cycles of amplification with increasing temperature. Walk-in nested PCR is made with different kDNA primer set. Low stringency cycle creates sites for prime annealing, and high stringency cycle augments the specificity of the amplification products. The PCR products from less amplification that hybridized with kDNA probe were cloned and sequenced commercially. The Chimera sequence shows the kDNA minicircle blue, intermediate microhomology recombination junction yellow, and the Gallus-Gallus DNA green. The mapping of the kDNA integrated in the chicken genome is obtained by a target primer TEL-PCR, TP-TEL-PCR technique. The kDNA mutated adult chickens develop Chagas heart disease. The mock controls grow to a healthy life. The kDNA mutated chickens show increased cardiac silhouette on X-ray films. The necropsies show control chickens have small heart size. The kDNA mutated chickens show hydropericardium, hydrothorax, and a large heart. The choriomegaly is similar to that in Chagas heart disease. The mock control chicken heart shows normal histology. The kDNA mutated chicken shows a tunnel made on the myocardium by the cytotoxic lymphocytes that lyses the heart fibers. The sequences of kDNA minicircles that integrate in code regions at several chromosomes are disclosed by a modified nested TEL-PCR that combines kDNA primers with host DNA specific primer sets. The kDNA mutated chickens die of heart failure and show choriomegaly. The microscopic pathology show myocarditis. The Chagas-like myocarditis, translating heart failure in kDNA mutated chicken, is a genetically driven disease. This is the first time that Chagas disease clinical and pathological manifestations are reproduced in a clean cross-kingdom model system. Thank you.